Very good morning to you all. आइडिया नहीं है कितने हैं, अराउंड 26 थे शायद, 26।
स्टूडेंट ही है या फैकल्टी भी है?
नहीं, मोस्टली स्टूडेंट्स हैं जो PhD। हाँ,
जो PhD में कुछ नहीं है। मेरा जूनियर आ रहा है एक तो यहाँ पे Geomics में था, जो मेरे को चश्मा लगाए रखता है।
कहाँ है
जी, ये येलो उसके जर्सी वाले के ऊपर में जो बैठा है चश्मे वाला,
कहाँ पे?
इधर ना दूसरे वाले लड़की एक सीता के पीछे वाली लड़की, सेकंड नंबर पे जो बैठा है इधर से चश्मे वाला।
कहाँ से?
मोदीपुरम से ना, सरदार वल्लभ भाई पटेल। एक एक आया होगा, एक एक कैंपस से।
हाँ, रिप्रेजेंटेशन
दो तो नहीं आए, सबको प्रेजेंटेशन भी है इनकी।
नहीं, मतलब हर एक जगह से रिप्रेजेंट किए होंगे ना।
हाँ, ऐसे एक एक दिया होगा। ये सब आया होगा, ये सिफारिश पर आया होगा।
सब तो यही चलता है।
बाकी सब कुछ कर रहे प्रोसेस है।
किसका वेट कर रहे हैं?
पता नहीं आया होगा कोई अभी।
पता ही नहीं है इसलिए।
यूट्यूब को देखो, मुझे ही करके रखते हैं।
Ladies and gentlemen, galaxy of intellectuals, the esteemed dignitaries, scientists, participants from across the country, and dear students, a very good morning to you all. On behalf of the organizing committee, I welcome you to the inaugural function of the NAHEP CARS-sponsored short-term training program on genomics for improvement of horticultural crops, organized by the Division of Vegetable Science, IARI. First of all, I take the privilege of welcoming the dignitaries on the dais, Dr. Trilochan Mahapatraji, the DG and Secretary DARE, Dr. R.C. Agarwal, DDG, Education, Professor Kole, Dr. Rashmi Agarwal, Dean and Joint Director Education, and Dr. T.K. Behra, Course Director. Before we go forward with the program, I kindly request our beloved director, Dr. A.K. Singh, to welcome the Honorable Dr. Trilochan Mahapatra, DG, ICR and Secretary DARE, the chief guest of today's function, with a plant.
Plant.
Plant, please.
Hey, plant first.
Plant ले आइए.
Plant.
कोई नहीं.
I request the dignitaries on the dais to start the program by lighting of the lamp. Thank you all. I now request our director, Dr. A.K. Singh, to welcome the Honorable Dr. Trilochan Mahapatra with a plant. I again request the director to welcome Dr. R.C. Agarwal, DDG Education, with a plant. I also request the director to welcome Professor Chitrajan Kole, Raja Ramana Fellow, with a plant. Thank you, sir. I now humbly invite Dr. A.K. Singh, Director IRI, to kindly deliver the welcome address.
A very good morning to each one of you. Honorable Chief Guest of today's function, Dr. Trilochan Mahapatra, Director General ICAR and Secretary DARE, Dr. Agarwal, Deputy Director General Education, Professor Kole, Professor Raja Ramanna Fellow, Dr. Rashmi Agarwal, Dean and Joint Director Education IRI, Dr. Behra, Coordinator of this program, Dr. Manjaya, Dr. Vishnathan, Dr. Saili, and all the distinguished faculty members from Division of Vegetable Science, and dear participants. It is my privilege to extend a warm welcome to our chief guest today, who is one of the most distinguished alumni of this institute. And the audience knows him so well that I really need not to tell the details, but for the sake of the participants who have come from far distances from every corner of the country to participate in this training program, I would like to tell you something which you might take as inspiration for your future career. Dr. Mahapatra did his graduation in agriculture from OUT Bhubaneswar, and then he did his master's and PhD from ICAR-IARI in biotechnology, in genetics, not in biotechnology specialization, but in division of genetics. And then he joined as a scientist in the National Research Center on Plant Biotechnology, and then rose to become principal scientist. During his scientific career of more than 3 decades at ICAR-IARI and RCPB, he made tremendous contribution, particularly in the area of genomics, molecular markers, molecular breeding, and genome sequencing. The outstanding research work got publication in journals like Nature, Plant Cell, and brought international recognition to this institute. I vividly remember when he was preparing the rice genome sequencing project, and he used to sit long hours in Ganga International Guest House with the then ADG, Dr. Dhillon, discussing about the project. I don't know how many times, and that discussion will always happen in the evening hours from 6 o'clock till 8 o'clock, 9 o'clock. And I happened to be a witness because I was in charge of the Ganga International Guest House, so I used to go there frequently. And the project took a shape, and it brought the country at an international map. As a teacher, the contribution that Dr. Mahapatra has made is phenomenal. His course on eukaryotic molecular genetics will always be overcrowded. There will be no space for people to sit. The classes will not last, less than 2.5 hours. Will always be extending beyond that, but still people will have the interest to listen to his presentation, not only from ICAR-IARI, but from other institutes, including All India Institute of Medical Sciences. Many doctors used to come to understand and know how the science, and this was the time when the science of molecular breeding was just progressing, markers were just progressing. I had a special privilege of the interactions with him because we stayed together as a next door neighbor in Rohini sector 15. And morning at 8:30 we will get into the chartered bus and travel to institute, and that was about 1 hour 15 minutes time, where he will teach me and we will discuss at length the things like AP-PCR, AFLP, the differences between them, SSR, ISSR. SNPs had not come by that time, but this discussion will go on for hours. Evening I will not have that opportunity because evening I used to leave 4:30 back in the chartered bus, but he will take a bus around 8:30 or so to go to Wazirpur Depot, I think 585 it was, and from Wazirpur Depot another change to sector 15 Dwarka, and will never reach before 9:30, 10 o'clock in the night. So you can imagine the tenacity and the commitment that he had to teaching program, to research program is phenomenal, and that is what keeps us guiding, motivating, and is motivation for each one of the students who are participating in the training program. There is no limit, and the background from which he comes, the area from which he comes, if you still, we had on some occasion we had to go to his place, and many of us come from rural background, but you can see the struggle for his school also. He had to go on cycle, maybe 30 kilometers cycling, crossing few 2-3 rivers on the way. So what I am trying to say is that there is no limit to the growth that you can make, provided you have commitment, perseverance, and dedication to your work, and that is what is testified by the life of Dr. Mahapatra. Welcome you, sir, for this program, and I am sure the students will be greatly motivated. I would also like to welcome Dr. Agrawal, our very dynamic Deputy Director General Education, who is also a distinguished alumni of IRI from the statistics. And he was Registrar General of Protection of Plant Varieties and Farmers' Rights Authority, wherein he brought several changes, many innovative changes, and harmonization of the laws internationally. And as DDG Education, he is providing great support to IRI. Soon after his joining, he came to me, and after I had taken over as director, and he came up with the proposal of almost 3.5 crores if we can modernize our library in terms of providing compactors, because space is very much limited, and how we can store more number of books. And since then, he is following every day what is the progress, whether tender has been floated or not, the technical specifications are decided or not. That kind of follow-up it is hard to find actually generally, but that's what Dr. Agrawal is. Thank you again, Dr. Agrawal, for being here. And he is the main mentor and pillar behind the NHEP CAST program that we are doing. Professor Kole is one person who is again an admiration for each one of them, and I envy him for his ability and sustenance to write books and edit books. The numbers runs into, I think, 141, because he keeps it updating on the Facebook also. The genome series in plants, large number of them he has done. Thank you, Professor Kole, for being here and for motivating our students during the training program. I also like to welcome Dr. Rasmi and my colleague Dr. Behra, who is very competent scientist, has got good international exposure in terms of vegetable molecular breeding, and he has made his presence felt at the international level as well through his publication and work. So I'm sure that this training will take all the students who have joined the training program to make them learn new areas and use molecular breeding in the field of improvement of vegetable crops. Thank you very much.
Thank you, sir. I now invite Dr. T.K. Behra, Professor of Division of Vegetable Science and Course Director, to give a brief introduction about the training.
Most respected Honorable DG and Secretary and DDG Education, Dr. Asya Agrawal, and Professor Chitranjan Kole, my teacher, and Dr. A.K. Singh sir, the director of this institute, and Rasmi Agrawal ma'am, the Dean and Joint Director of Education of this premier institute. Head of the divisions and Madam Sally and Dr. Vishwanathan, Dr. S.K. Singh, professors, scientists, faculties of all horticulture, the discipline is there, because this topic says it is in genomics for improvement of horticulture crops. So this is the students' training. So we have many of the students come from the different places, and as we know that we have a really privilege that we have all the persons from the genomics side, the genomics and also the genetic resources, which is important for our program. And for horticulture crops, as you mentioned, just some of the things like in horticulture, sir, in India, we don't have any progress in horticulture in the genomics sense of point of view. But if you say that is genomic data, if what we have generated, that is there throughout the world, if you see there are 181 horticulture species already sequenced. And 181, and only we are only in the tomato genome at the part, and other things already been sequenced throughout this country. And in vegetables we have 42, and horticulture fruits we have 64. And also in plantations crops, then spices and floriculture crops, we have data is there in this world as a whole, and it is there. But if you say the application part also, we have not done so much of these things. That's why we have to have a lot of works in these horticulture crops, particularly for the basic science and other things will concern. So this is not that only the training for inaugurations, but it is also in the group of horticulture scientists and other people of the students who are working. We have to work for the genomics and basic science, and that's why we have organized, and we have a panel of people, dignitaries who are here, and really we are really pride that it is there. We are having some of the works we can do in the future with respect to this one. So this is the starting point for us and for the horticulture group as a whole, and we can also take all the tools and techniques from the different disciplines and we can definitely develop the good infrastructure as well as the genomic resources particularly concerned. So coming to this program, sir, we have 89 students and applied from the different universities. That 23 universities have applied 89 students, and out of which we have selected only 27 students because we have to have only 25, but we have selected 27 because there are a lot of people also want to have the students want, because they are very much interested for this particular to come to the IRI as to have the work and also to get some knowledge with respect to the different genomic resources. And we also have the program in which we have selected these students. Those are working in the genetics particularly, not they are working in particularly in the genomics approaches, but still they have a very good knowledge in the genetics, and they are also taking the different minors in this case. In that case, we have 27 from 14. We have 14 states: Punjab, Uttar Pradesh, West Bengal, Jharkhand, Andhra Pradesh, Kerala, Delhi, Jammu and Kashmir, Gujarat, then Bihar, and we have also student from Arunachal Pradesh. So these are the states from which about 16 university students are there. So totally will be 16 universities and 40 states. So I think they are most welcome to these premier institutes, and they have a dream. They are already mentioning in the social website also that we have to come here and have to work to get some knowledge in respect to the horticulture sciences concerned as well as genomic sciences concerned. So I must welcome all these students here. I think, and we have about 29 classes and about 19 practicals, and all the classes will be taken with the different other sister institute, including NBPGR, then NIPB, as well as NIPGR. And also we have the most of the work of these classes will be taken by these genetics divisions, where we also have the opportunity to have students there in these genetics divisions, and we also definitely have all the courses you can see which is planned here, starting from that of this advances in different crops itself to the next generation sequencing. Even for the genome editing, we have also programs there. So all things we have categorized and put it in this programs and schedule. So I think it will be all the students will definitely get succeed for that. Thank you very much, sir.
Thank you, sir. I now request the student participants who have come from across the country to introduce themselves, giving a brief about their university, state, and research work. You can give your name, your university, state, and the work in which you're doing.
I'm Bipin from Tamil Nadu, and I did my UG from TNAU. I did my UG.
You're audible, go ahead.
I did my UG from TNAU, and
You speak something.
I did my UG from TNAU, and MSc from Uttarakhand University of Horticultural Science and Forestry, and I am doing PhD from Floriculture Division IARI, and I am working on salt tolerance. That means related to
Vikananda Agriculture Institute, Jodhpur, Jaipur. MSc from Manavta Institute of Agriculture and Technology, Udaipur. Now I am studying PhD from Visa Agriculture Institute, Jharkhand, and working on the diversity mapping of Jaipur for nutritional values in Jharkhand.
Good morning to all of you. Myself, Mujib Ahmed. I belong to the state Rajasthan. I completed UG from Bandar University of Bandar and completed post-graduation from Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut. Now pursuing PhD from Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut. Good morning, sir. Myself, Pito Kaur, UG completed from Bandar University of Agriculture and Technology, Meerut, and PG completed from Narendra University of Agriculture and Technology, Meerut. And now pursuing PhD Vegetable Science in the Higher Agricultural University of Bhagalpur. And my research crop in breeding tomatoes for quality with the
My individual program was from and post-graduate program was ICAR-IARI University of Agriculture Technology, Maharashtra, and now I am pursuing PhD in agriculture from Maharashtra University. My research work is related to physical attributes of field development.
From Varanasi. I have completed my UG in the Autonomous College Varanasi. Now I am pursuing PhD in vegetable science. My research topic is assessing genetic diversity of population in Assam.
सर एक वो वाला माइक भी मैंने दिया था, वो पास ही नहीं हो रहा। पास करना है बच्चे, उसमें बोले कि तभी तो कैमरे में ऑडियो आएगी, मैंने इसके लिए दिया था आगे।
I'm currently working on in vitro mutagenesis and validation of mutants in genome management.
Good morning, everyone. I am from
वॉइस में आएगा
ये खाली कैमरे के ऑडियो के लिए,
तो सुनाई भी नहीं देगा ना।
कैमरे में तो आएगा।
And I am doing my PhD
वो तो बात तो
from
सीख के थोड़ी बना
Sugar and Vegetable Science. And my research topic is morphogenetic and biochemical characterization of small size potato tubers and production of pathogen-free potato tubers through in vitro propagation.
Good morning, everyone. My name is Dr. Thomas. I'm from Arunachal Pradesh. I'm doing MSc in Medicinal Science. My topic is genetic variability in leaf mustard. That's classical example.
Good morning. Myself, Atilia. I have completed my UG from Kerala Agriculture University, PG from Tamil Nadu Agriculture University, and now I am pursuing my PhD from Department of Vegetable Science, Kerala Agriculture University. And I am working on genetic improvement of horticultural traits of bacterial-resistant tomato varieties. Good morning, everyone. Myself, Vidya from Tamil Nadu Agriculture University. Now I am doing second PhD in the Department of Spices and Plantation. Cultures for quality improvement. Thank you. Good morning, sir. My name is Megha Raghavan. I'm from Kerala. I did my UG from Bhagalpur, PG from CAU, Imphal, and now I'm pursuing PhD from CAU Arunachal Pradesh campus. My work is on genetic diversity of citrus reticulata using HSR markers. Good morning, respected teachers. Myself Krishnaja. I'm coming from Kerala, and I pursued my graduation from UNMT in Maharashtra, and my post-graduation from DBS in Dapoli. Now I am doing my PhD degree in the crop drumstick in the University of Horticultural Science, Bhagalpur, and I am working on the different aspects like morphological and molecular characterization for genotypes, its nutritional improvement, and the tissue culture protocols. Thank you.
Myself, Navina from Tamil Nadu. I did my UG and PG in PNA Coimbatore, Tamil Nadu, and currently I am pursuing third PhD in Department of Horticulture and Landscape Architecture in PNA Coimbatore. My research work is on induced mutagenesis in hibiscus for enhancing ornamental as well as the commercial value.
Myself, J. Purushottam from Tamil Nadu Agriculture University. Currently, I am doing a science PhD in Department of Vegetable Science. My research area is breeding for TLCV and NAMDO resistance through multivalent advanced generation intergas in domain.
Good morning to all the dignitaries. Myself Srikant. I am from Dr. YSR Horticulture University, Andhra Pradesh. I did my undergraduation and post-graduation from Dr. YSR Horticulture University. Now I am pursuing my PhD, and my research topic is genetic analysis and validation of markers linked to powdery mildew resistant and other traits in chili. Thank you.
Good morning, myself Prashant Sigulpati from
Back
TNAU.
दे दो
UG I did from TNAU and
देखिए खुला हुआ बैग
second
खुला हुआ
chemist. My
उल्टा
research topic
बैग
is
खुला
genetic
है
emergent studies in garden
बैग की चेन खुली हुई है
biomolecular chemistry.
Good morning to all the dignitaries. Myself Anamika Upadhyay and I am from West Bengal. I have completed my UG from City University and MSc degree from Inauguration Vishwavidyalaya, and at present I am in second year PhD second year Vishwavidyalaya West Bengal. And my topic is on characterization of fungal genotypes using with special emphasis on purple brush disease using molecular methods. Thank you.
A very good morning to honorable dignitaries, teachers and participants. My name is Rajni Rajan. I am from Bhagalpur, Bihar. I did my UG from Acharya Anjiranga Agricultural University, Hyderabad. I pursued my PG from Navsari Agricultural University in Gujarat. And recently I am pursuing my PhD from Bihar Agricultural University in fruit science. And currently I am working on in vitro mutagenesis in banana cultivar grain 9 for its improvement against salinity, with the major objective of generation of mutant lines and the selection of some lines resistant to salinity. Thank you.
Good morning everyone. I am Neha Varma from Himachal Pradesh. I have done my BSc and MSc from Dr. Y.S. Parmar University of Horticulture and Forestry, Nauni, Solan. And at present I am pursuing my PhD from Punjab Agriculture University in the Department of Vegetable Science. And my research work is on inheritance and mapping of yellow wing mosaic virus disease resistance in pumpkins. Thank you.
Good morning to all. I am Anisha. I am from Jammu and Kashmir district, Jammu. I have completed my BSc and MSc from SCOS, Jammu. Now I am pursuing PhD from Punjab Agriculture University, Ludhiana. And I worked on marker-assisted breeding for inauguration of genetic maize and changing from phosphate to tolerant bipeptide.
Good morning, one and all. I am Ipsita from Odisha. I did my UG Odisha University of Agriculture Technology. Then I have joined Chaudhry Charan Singh Haryana Agriculture University for my postgraduate. Now I am doing my PhD in Division of Vegetable Science, and I am working on QC mapping for regulatory traits and vegetables.
Thank you, dear students. I now invite Professor Chitrajan Kohli to say a few words.
Dear distinguished dignitaries on the dais, specifically Director General ICAR, Professor Mahapatra, Dr. Agarwal, a dynamic person leading in agricultural extension, Dr. A.K. Singh, Director IRI, Dr. Agarwal, Dr. Behra. We all know that the concept of genome, the word genome came in 1928 and it was coined by Dr. Winkler. And the term genomics came in 1986. And during that period actually, molecular map construction, mapping of genes and QTL started. But to be very honest, in India, genomics started in the 90s. And that time, most of the people were busy in genetic transformation. So in biotechnology, most of the scientists were engaged in genetic transformation. But it is a proud privilege to tell that one person who is in the dais today started one pioneering work in genomics, that is construction of genome library in chickpea. He that time was working with Professor V.L. Chopra, another legendary scientist in India. And later on, he continued his molecular mapping works in Indian mustard. And the first molecular map from Indian soil was developed by him. It was maybe 1995, if I am not wrong, isn't it? So then he went on to map genes for resistance to biotic stresses. If I am not wrong, he was mapping white rust resistance, albugo candida in Indian mustard, and then followed by QTL mapping. So you have that pioneering plant genomics person today on the dais, and a lot of things already have been enunciated by Professor A.K. Singh, present director of ICAR-IARI, so I don't like to elaborate. But one thing I must mention, that application of genomics, hardcore genomics, started in the early part of the century, precisely after publication of Rhodopsis genome sequence. And it was followed by rice, the first crop plant to be sequenced. And again, Professor Mohapatra was a party to that. This paper was published in Nature, as Dr. Singh told. And he continued, and this continued, and always he was included in the teams of genome sequencing. I think tomato was followed by that. I don't like to elaborate. So, and I believe ICAR-IARI is the premier institute in the world, not only in India. It has led green revolution in the total developing countries in the world, and I believe using genomics in all types of crops, agronomic and horticulture, will lead in evergreen revolution. Thank you very much.
Thank you, sir. I kindly request Dr. R.C. Agarwal, the DDG Education, to address the gathering.
Honorable Secretary DARE and DG ICAR, Dr. Sudarshan Mahapatra ji, Dr. A.K. Singh, Director of this esteemed institute, Dr. Kole, a renowned scientist, Dr. Behra, Dr. Rasmi Agrawal, my colleagues from IARI, senior faculty, and dear participants. I think for the students, you should be proud that you are attending this important training program on important topic from the important institute. It's really a lifetime opportunity that you get something from IARI. So I compliment each of the participants that they have been selected out of a big number, keeping in mind the social equity, keeping in mind the gender balance, which I could see. Dear students, I think again it's a matter of pride for you that you have the great visionaries sitting on the dais. You can have, as Dr. A.K. Singh has spoken, Dr. A.K. Singh himself is a great source of inspiration. The kind of life he was narrating for Dr. Mahapatra, I think his life is also the same struggling life, which I think you may know at some other platform, but not today. But you can certainly know that how he struggled and how he has reached to this position, a prestigious position, which is a dream for all. So with that small note, let me just tell you that under this NAHEP CAST program, this is a regular phenomena by this institute, and whenever I come for attending such programs, not all programs, 2-3 programs I have attended, I can see that from a very big number they are selecting. There is a huge competition. Like for this also they were saying 90 applications were there. So it's a very fair selection, and they always keep in mind every kind of thing, like there is a suitable representation from all these states which we are seeing today. There is suitable representation they keep in mind this gender balance also. Okay, and a special provision which we have in our NAHEP as such, because it's there are 3 subcomponents. So in each subcomponent we always say that you have to keep in mind the social equity. So we have the equity action plan, So that also is kept into mind. Sir, to DG sir, I would like to just say that under NEHEP, we have a target of around 90,000 persons to be trained. It's a big number. And recently this number was reviewed by World Bank, and till January we have achieved a target of around 24,000. We have an online tracking system that is called Project Monitoring and Tracking System, where on day-to-day basis all the partners, they have to update the various parameters, various indicators. We have around 62 indicators, and how many beneficiaries are there, this is also one of the indicators, like placement ratio, okay, like the girl students, like how many students are going abroad. So there are many parameters, and in all the parameters we have the base values, and periodically whenever the report is submitted to the World Bank or to ICAR, we take help only of the PMDS. So under that, I think IARI is contributing a lot for training the human resource, for bringing the human resource to make the challenges, to meet the challenges in the future. I think that's a great effort of this great institute, and I should compliment the director and the whole staff that they regularly, I think once in a month I could see the posters at the gate that there is some program of INHEP going on. So that's really something great. Now friends, the project which is under which you are attending this program, it is a very prestigious project. The total budget is around 1100 crores. But the kind of opportunities which are being given to the students, I think it has never happened in the past. In future, I don't know how we are going to sustain this, but we'll make some plan for the future also. Where even undergraduate students are getting opportunity to go abroad for 3 months, 6 months at the cost of 10 to 20 lakhs. So that's really a lifetime opportunity, and these opportunities has to be availed by the students. Every student cannot go abroad because of many reasons, because of the limited resources. But a student who is going abroad, or a student who is going to the best of the institute for his internship, those best of the institutes may be within India also. So they have to really give this message to other students, and have to motivate other students also that what are the best opportunities in agriculture, how best you can contribute for the society, how best you can meet the challenges of the future by availing these facilities. And sir, one more thing I would like to just inform. Last year there was an increase of 60% applicants for appearing in this All India exams, entrance exams for the agriculture. 15% seats of the UG are being filled up through ICAR quota, and 25% in MSc and PhD. So there was a 60% increase. Now this year I have asked all my partners, 46 partners, 46 agriculture universities, that please conduct a workshop during the month of February. Already around 20 workshops have been completed, by calling school principals, science teachers, and by calling parents, so whosoever wants to come, and just explain what are the different opportunities in agriculture. And through my analysis, I have just found out the only reason why students are not motivated towards agriculture is because they are not aware of the opportunities, the facilities which are there. And once they are explained, they get so excited to appear for this, and they want, once they are explained, they want to come to agriculture by choice. So by choice, you must have seen after 12th class, most of the students, they may be either preparing for medical or engineering. But hardly we find that after 12th class, they are preparing for agriculture. So once they are not getting somewhere, then they will think of, okay, what are the other options? But that attitude we want to change. And we want the best of the best brains towards the agriculture. And that's what we are making the efforts through this project. Sir, we are, I have already compiled a list of all the universities where the IPR cells are there, and accordingly we are going to have very strong IPR cells in all the universities. And similarly, I have made a concept note for making a placement cell also in all the universities, so that there is a networking. I was asked by Madam Purvi Mehta during my recent visits that she wanted some 3-4 persons of say particular discipline, but she was saying she could not get. Everybody was saying you contact this university, they were saying no, you contact that university. So there was no networking of all the placement cells, no uniformity in the procedure of the placement cells. So that also now we are trying to target, that how in all universities we should have uniform guidelines, we should have a networking of all placement cells. So if somebody approaches us as a DDG education, we should immediately flash it through that network that okay, this is the requirement, and if somebody knows, they can. The third thing, sir, which we are going to do within this month and next month, is the creation of alumni network. And already some 10 universities under this NIHL, they have conducted a big workshop of all the alumni, and many universities are saying that the alumni who are well placed, who are entrepreneurs, they are coming forward to support the university in various activities. And that is what beyond any HEP we want. So somebody is just trying to sponsor some buildings, some hostels, they want to sponsor some students. So that also for the sustainability we are trying to make through this project, that how the alumni network is there. And at the central level, we are building a software through which there will be an alumni network of all the universities. And the uniform software will be used by all the universities for this particular purpose. So there are many other things which we are trying to do, but I'll not explain all those things, but only compliment each one of you and thank our head of the family, Dr. Mahapatraji, that he is giving a new direction for attracting youths through education division, and to Dr. A.K. Singh, that I can see a new energy is flowing in IARI after he has joined. In a short span of time, a lot of new things he has done. And I also propose certain changes in the academic council meeting. So let us hope that for the betterment of the students, we come up, we work jointly, and can further bring the standards of the education, agriculture education, at a high level, which is the motto of any HEP, to bring the agriculture quality at a very high level and to bring it more relevant for the social cause. So thank you once again, and my compliments again to the students for getting selected for this training program, and all the best for the students. Thank you.
Thank you, sir. Now is the time for the release of the book, so I kindly request the chief guest, Dr. Trilochan Mahapatra, to kindly release the two books, and I request the other dignitaries to join. The first book is the Training Manual on Genomics for Improvement of Horticultural Crops, edited by Dr. T.K. Behra, Dr. Amit Kumar Goswami, and Dr. Gojrat Singh Chand. The second book is The Bitter God Genome, edited by Professor Chitrajan Kole, Dr. Hideo Matsumura, and Dr. T.K. Behra. you all. I now invite our Honorable DG ICAR-IARI Secretary Dear Dr. Trilochan Mahapatra to share his few words of wisdom to the august gathering.
Good morning, everybody. Dr. Agarwal, DDG Education, Dr. A.K. Singh, Director of prestigious Indian Agriculture Research Institute, Professor Chittaranjan Kole, Dr. Raja Ramanna Fellow, and also ex-Vice Chancellor of Vidhan Chandra Krishi Vishwavidyalaya, Dr. Rashmi Agarwal, Dr. Tushar Kanti Behra, colleagues of the dais, faculty, students, and particularly the trainees who have come from outside. A very hearty welcome on my own personal behalf, on behalf of the Indian Council of Agriculture Research, to each one of you. At the outset, let me thank Director IARI for inviting me. In fact, Dr. Vishwanathan, who is PI of the CAS project, he contacted me, and subsequently Dr. Behra also, and then Dr. A.K. Singh, Director himself. So though I didn't have this scheduled program in my engagements today, I had other things, I had to postpone that. Then I thought I will come here. Earlier also I came, another occasion, in this CAS program. Because I am here nearby, and I am also, as it was said, I have been a student of IRI and continue to be a student. So that learning process continues. So I try to find time, and today I am here. More importantly, to know each one of you who have come from different places to participate in this training, CAS training program on genomics for improvement of horticultural crops. You are fortunate today that you have teachers to teach what you are expected to learn. And those days when we initiated, so there are no teachers actually to teach and solve the problems that we were facing. We had teachers who did teach us, the fundamentals of molecular biology and DNA techniques. Professor S.L. Mehta, who then rose to become the Deputy Director General, was Dean here and Deputy Director General Education. And also Professor K.R. Kondal, who subsequently became Director of the National Research Center for Plant Biotechnology, and Dr. I.M. Santa, who was also a principal scientist and professor in biochemistry. And subsequently, my own teachers, Professor Chopra, Professor R.P. Sharma, at the National Research Center for Plant Biotechnology. So they are the teachers who taught us and gave us the fundamentals of how to really do things. But when you go inside, as Professor Kole said, 1986 is the watershed year. In 85, the linkage map concept was published in Human. And in 86, the papers appeared in Tomato and Maize. And first of all, conceptualizing map construction. And then subsequently, the maps appeared. And that time was the starting point. And in 87, when I was planning for my own PhD program for a similar work, And though we knew that how to isolate DNA, but isolation from brassica young leaves took me 6 months to standardize, to get good quality of DNA. And those days were the days of ultracentrifugation to get purified DNA. And hundreds of DNA samples were put to ultracentrifugation to get pure DNA for southern blot hybridization. And to get results, it takes 15 days, at least a week. So, and all radioisotopes sitting behind me and then in front of me for 4 years. So those are the kind of situations when you have to struggle. And we had to identify low copy sequences for hybridization. And we didn't know actually how to really get it. And I read quite a bit of literature. I started planning how to get it. We started dot blotting DNA. And you would not imagine the specifics that we had to really design ourselves to get a wonderful dot blot. And they were even better than what was there then in the textbooks and global level. And that's the kind of southern blot and dot blots which we generated. So Professor Deepak Pental was those days in Terry. He came back from US, and after many years of postdoctoral program and PhD in the UK, and he looked at my thesis and they said, "You work here or in US?" So I don't believe that you can do such kind of things here. So the struggle that we went through ourselves, southern blot took another 6 months to be standardized. We're not getting anything in chickpea, and then I moved on to brassica, and then we succeeded. And then that is how, then subsequently chickpea also worked. And these two crops where we focused on. So why I'm telling this, that when you don't have anybody around to give those kind of inputs, as I said, we had teachers to give those fundamentals, but the support with regard to specific requirements for developing molecular marker system was not there actually in 80s in India. Everybody was trying to do something, but there were the problems. But anyway, so what I was trying to say and emphasize, that whatever we learn, whatever you learn through these struggles, trials, that make you the best in the sense that you don't have to depend on anybody, and you develop a mastery over that. So that is the kind of point, and that is what I would tell at the outset, that all the trainees that you have come from different institutions, universities, and in fact those who are working here, this is the point, borrowed ideas and all that would not take you farther. So you have to have your own ideas while working on the ideas of others. And that is how you are established as individual scientist, having your own strength. And Dr. Kole was telling, Dr. A.K. Singh said that we were discussing in bus and all that. But the kind of his own idea Dr. A.K. Singh has put in, that is unparalleled. It is not just you can get some idea from somebody and then you will click. That would not happen. May happen once, not more than that. So it's Dr. A.K. Singh who has put in own idea into it and then developed the whole field of translational aspects of it. So he's an example. So likewise, when I was doing QTL mapping, so I went to ICAR-IARI, there was nobody actually who worked in this particular area. So there are difficulties to explain actually what I want actually. How do we do QTL mapping? That was, late 80s. And the first seminar when I delivered on QTL mapping, not many people actually believed that QTL can be mapped. And the people thought that they're all abstract things. And when we say mapping, it has to be physical. So when there is nothing physical, so how do you map actually? And even the quantitative genetics teacher those days was a brilliant teacher. In fact, did not really believe this, that it can be really mapped. And then subsequently, when I talked about cloning of those, and that was again another hard area to actually believe that it can be cloned. And those days, not a single gene was a clone actually. So when I talked, through schemes, and I developed schemes myself on how to do marker-assisted breeding when I used to teach students. So, you didn't have anything in the net and nowhere. There were only some papers here and there. And my initial papers of QTL mapping was based on the isogynes, not actually DNA markers. Published in Genetics and Crop Science, and then Weber, and then others. So those papers, they were really guideposts, on QTL mapping. So you derive concepts from somewhere and then try to develop some schemes and how to really go for marker-assisted breeding and pyramiding. Pyramiding, which I used to teach, and I made a slide on those, how we can pyramid genes those days. And I started taking classes in 89, 90, I remember, when I was still a student. And I used to teach this, and started in 92 when I came back as a scientist, I started teaching. So the lessons from this is that when you trade a path alone, it's the toughest thing to do, but you learn so much. But you have to have courage to trade the path alone. And that is how you also make the path for others. And that is a point which is very hard to do, but then it's very satisfying to do that. When I was isolating DNA, plasmid DNA for southern blot hybridization and all that, hundreds and thousands of them were there. So I had to spend 36 hours at a stretch. I didn't go for anything, so I was there in the lab. So that's the kind of determination you have to move in the direction that you want to move actually. And you think that you'll have 3-4 hours of work and then so many other things to do, and you can do something, but you can't. You give 100%, then you get the results as you expect. So my suggestion would be that you have come for training, and the colleagues would tell you how to do DNA isolation. I was looking at those DNA isolation, all those things, and sequencing. But that's not really enough. That's not really enough. Training is just to expose you. You have been already working, some of you, in this area. But there are many things beyond that. And that you have to actually aspire for. And how you aspire, that depends on individuals, and then certainly it will define what you become. When I was trying to do QTL mapping, so as I said, my ISRI experience didn't get much. There was one Dr. Prem Narayan who used to be in the central office, so who was also director of ISRI and all that. So tried to explain me with all derivations and then the single factor analysis of variance. And you should be told how it is done. Today we go for some software, put the data, and then get some results, and then we don't know what we are getting. We don't understand the fundamentals. And we have 10 markers, and then you know how many pair of interactions you will get. You get something, thousands and thousands of interactions, and you start writing your thesis without understanding how many biological interactions are possible actually based on 10 loci, if you assume that. That is what students start doing, and that should not be done. So try to understand the fundamentals of it. Not easy. It requires a bit of background of statistics and mathematics. But if you want to really be in this area, you must understand. And Dr. Kohli was back those days from US, and he came to NRC plant biotechnology. In that sense, he showed me how to use QTL map in Map Maker module to do QTL mapping. Using Map Maker, particularly interval mapping. So that is how you pick up, single marker analysis, single factor ANOVA, and then QTL mapping using intervals. And subsequently, we did composite interval mapping. And those papers which we published those days from India, they were the first ones actually. Dr. Kohli said map and subsequently the QTL ones, they were the first ones from the country. Not often actually cited, because we published in Indian journals, they are not often cited, but they are the first ones from the country. They were very tough ones because southern hybridization, as I said, kind of long procedures that you have to follow. So friends, you keep your minds, eyes, ears open to learn from everybody. That is the best part. And acquire from everybody, from your friends, from your teachers, from the books, from journals, from, I don't know how many of you go to library nowadays. That's another part. Whenever I come here, I take this opportunity to tell you that my best learning happened during MSc. Not PhD. PhD I started working and then day in and day out I will be there in the lab. But best part was MSc. That time I would go to library and then sit there and learn, Cell gives mini reviews. They're toughest ones to actually understand because unless you read previous 10, 20 papers, you will not understand what they are actually talking about. And when you do that, in the process I collected huge literature. And those days, my professor, you have some money given for this purpose. A professor used to question, what are you doing all this? Xeroxing so many. Every time Xerox bills will be submitted to, professor for approval. So he used to question me. So that gives a very broad understanding. So I hope you will have that kind of interest. Acquire from every source and read the horizontal papers. Otherwise, you will not develop that kind of background. As I said, by borrowing, you can't sustain for a long time. So this is what I would say. Genomics for improvement of horticultural crops. So when we started tomato genomics in this country, nobody did, anything. We didn't know ourselves how to do genomics research. We used to teach something very basic in the class. So without that, we ventured into it and got about 50 crores those days in 99, 2000. And that was first of its kind project in the country. So, you will get support, but then you have to have firm faith in what you are doing. And unless you have that faith, nobody is going to support you. And you have to confidently say that you will deliver without knowing anything, without doing anything in that area. We were telling confidently that we will do it. So that is the kind of thing. That is how genomics started, rice genome started, and subsequently tomato genome. And then, our labs in NRCPB were open to all the people at IRI, and of course horticulture was open. And that is how my name is there in rose, in a mango, in Karela, and maybe 20 crops of horticulture. Not less than that. So, that's also another part of it, that if you are too possessive, you don't contribute much. Maybe you will publish few papers and then get some awards, that's okay. But if you are open, you build a whole world for yourself and for the nation. And that is the another point I would like to say from my own experience, that if you are open, that you build a whole world for yourself. And if you are narrow, clumsy, and then you have a very small world, either you want a small world for yourself or a very big world for yourself. So that you can determine. And that's another aspect of your development. I think you will take care. So time has gone, and then I think I have spoken for 20 minutes. But coming to genomics per se, I'm sure you will be learning quite a bit. Say structural genomics component and functional genomics component. There are two fundamental aspects to this. When you say that you have to sequence genome and you have to develop some tools for further use in breeding for application, there are many different applications from structural genomics. When we started structural genomics, was initiated in the form of developing saturated genome maps. And you can ask this question, what is a saturated genome map? Saturation needs to be really defined. And I used to define in the classes, but I believe you will be told about it. But anyway, so the saturated genome maps. And subsequently, sequencing base by base the whole genome, and you have everything there. All the features, the structural features are there before you for the whole genome. So once that is available, that's a big tool. Tool for designing markers, tool for designing tools for functional genomics, tools for doing even transgenics. So all these follow from that whole genome sequence analysis. And this is what is actually the starting point. But that's not the end point. That's what many people believe, that if you sequence the genome, everything is done. Actually, this is just a precursor to do many things subsequently. And in fact, in this country, we started genome sequencing, continued sequencing many genomes, wheat and participating wheat, genome sequencing, chickpea, pigeon pea, so on and so forth. And then even we have sequenced by now mango, jute and other crops. So this is continuing. And then we started taking advantage of this in rice. Tremendously we have taken advantage. And that is what I was telling Dr. A.K. Singh, your own director, applying a mind to translational part of it and then developing that whole area. But in other crops, we haven't really done much. I was compiling total varieties we developed through marker-assisted breeding. And this was totally about 48 of them we have developed. And when we did the first, we published in developed and released in 2007 in rice. And subsequent to that, about 46 of them have been developed. So, and you find majority of them are in rice. And then followed by wheat and maize. And other crops, maybe one or two examples. In the horticulture, probably there is no example of marker-assisted breeding to develop a variety in horticulture. So in that sense, when we say that we have so many genome sequence, so quickly these trainees should be told how to develop markers out of this sequence, validate them. We have so many papers published on this, and then immediately put to use. And if they can learn that, they can actually go to these genome sequences and then get direct benefit, immediate benefit out of it. Those days we used to start struggling with RFLP, then RAPD, and then AFLP, and we went on. Actually, in evolutionary terms, if you see, myself started with RFLP, and then in fact, did some isozyme as well, and then moved on to SNP. So, this is a kind of perspective that has brought. But then, what is important for you is, designing that kind of markers. Dr. Cole said that they are highly heterozygous and so on and so forth. And if you have codominant markers, you don't care for heterozygosity. And SNPs are codominant. And so also the microsatellites. And I'm sure you can derive maximum benefit out of these genome sequences. And then, design your own markers, choose a crop appropriately, and then move on. And some of you are doing PhD, and I know now itself you can take advantage of that information which is available. So this is one aspect of it. The other is that if you are believing and more interested in the fundamentals, there's so much to do from the genome sequence. So genomics in entirety, I do not know how you are going to be exposed to, whether it is only translational component or there are some fundamental aspects. So understanding the structural variations there. Tremendous structural variations are there. From evolutionary viewpoint, how relevant they are. From crop application, even translational aspect, how important they are. Not only in the coding part of it, the non-coding part of it. In fact, we published a paper on non-coding, microsatellites, and how important they are. And similarly, how important they are even in repetitive regions of the genome. We often believe they are junk, but they play so important roles. Even the mobile elements. So I think the entirety, if you study the genome structure itself and try to understand what it is, I believe they will be told about it, and they can go with a much better information about it. And beyond that, somebody said you were going to do transcriptomics and then so on and so forth, and we did that, almost 10 years ago, and people now still continue doing that. And it gives a whole lot of information. Whether you make contigs or you go by ESTs and all that, and then you do comparisons, identify genes which should be expected. But that is again the starting point of another huge work which would be awaiting you. You'll get 1,000 genes upregulated, 2,000, 5,000 downregulated, and do some pathway analysis, and then find out, maybe another 100 genes and 200 genes to actually to deal with. Not easy. So you must know how to really narrow down to few and then move beyond. But all these genomics and transcriptomics and genome variation at the global level when you do multiple genome sequences of different varieties. So all these have to again relate to your phenotype. So unless you do phenotyping appropriately, you will not really get much of results in translation. But I must tell you one thing here in phenotyping, that gross phenotyping mostly we confine to. But if we talk about Karela, anti-diabetic property, and so on and so forth, and maybe there will be a quantitative nature to the compound which is responsible for this. And if you do that, you have to go down to metabolite profiles. Unless you do phenotyping at the metabolite level, you will not make any sense out of your application using this transcriptome and then genome informations. So what I'm trying to tell you, that when you talk of genomics, I used to put everything under genomics, whether it is functional and functional component, even metabolism, metabolite profiling as a functional component of it. So, though some people try to put them separately, but without those information, this information is incomplete. So, all this information together, the phenome, the genome, the metabolome, transcriptome, all together, and you can't do everything. So obviously, you can imagine the complexities which are lying there in this genomics for improvement of crop plants. If you start with a marker-assisted breeding, that would be the simplest one, then a simply inherited trait and one gene, major gene, and you have one marker tightly linked, or the gene is cloned, gene-based functional marker, use marker-assisted breeding, fine. When you talk of QTL, then you have another layer of complexity coming in, and you have a linked marker, but you don't know, as you know, the QTLs expressed differently in different environments. You have epigenome and its control coming in, and epigenome is another complex area, another layer attached to that. You don't know how to really deal with this. So the whole area is full of challenges. The whole area is full of challenges. and more so horticultural crops, because the perennial nature of these crops often creates many layers of complexities, because there are different seasons, and then there are different on developmental phases, and then long periods. So as Dr. Kole said, so you have further layers of complexities there, and to understand that, and epigenome variations in perennials, very little understanding. Actually, we don't understand. We don't understand actually. Biology is very poorly understood. Very, very poorly understood. The most poorly understood area is biology, despite all the developments. Genome sequence doesn't give much, I can tell you. So that's the kind of scenario which is there. But sequencing, it doesn't mean that you don't sequence. So unless you sequence, you will not understand the genomes to start with. So friends, I said the human side of it to start with, that you have to struggle, you have to strive, you have to aspire, you have to work alone. Even if you have to work alone, not necessarily you have to work alone, isolate yourself from others. If it requires to work alone, and obviously if you want to be a pioneer, you have to be alone. If there are many people, obviously your journey would be very happy journey, very relaxing journey. There are many people with you, and then certainly you will be moving a bit slowly, because you'll be chit-chatting, you'll be talking to them, you are learning from them. So, but it will be a very enjoyable journey because you have others with you. But if you are alone and you are trying to explore something which nobody else is doing, then obviously, as I said, it will be the very, very tough journey. Walking alone is very tough. You might walk fast. So that is what I said in the beginning. But I'm sure all of you would do that. And you have teachers now. So teachers to guide you, to handhold you, and then tell you. But if you want to go beyond what teachers are telling, then you have to struggle alone. That is the point which I wanted to tell you. So are you prepared for that? I don't know how many of you are prepared for that. So I'm sure, as teachers, our job is to inspire you. We were inspired by our teachers. And I always say that today's students are better than the teachers of today. So that's the reason why I say we have a bright future. If the students of today are no better than the teachers, we have a, very, very, I would not use harsh words, difficult future ahead. So I am sure you are all better than, more bright than each one of us, and each one of your teachers with, more ideas. You are in the digital era, and digital era provides immense opportunities to be captured and also to be taken advantage of. Not to waste your time, energy, and also mind. That's very dangerous. Waste your mind also sometimes. Get bogged down by something because of the digital platforms, because you become attached. So that is what the technology offers now, and that time we didn't have. And that was also one kind of a distraction, if I see today. And they were not there, so we were one way focused that way. So you have to fight that distraction and stand there. So I hope you will do that, and you will certainly do better than the teachers, and you will explore new areas. Just learning under this CAST program should be a very small component of your total learning. And you have to go beyond that. So this is a bit philosophical, but I'm sure you will be able to do that. And I also told you genomics in entirety, the complexity of it, and don't start doing everything together. So start step by step, and I'm sure you will succeed. You have to have a very good plan of action before you read thoroughly. If you don't do literature survey, you will be telling only in your thesis that you repeat what was said 20 years ago. And your results are confirmed, are supported by something which was done 20 years ago. So what is that new you have done? Is something which was done 20 years ago is supporting what you are doing today. So obviously you haven't done anything new. So our whole thesis will be full of this kind of discussion and interpretations. What is new and how do you put that in a proper perspective? That you must learn. My own students, our own students from ICAR-IARI and students from various institutions and universities, this is what is very, very important. And when you try to write your even review of literature, copy para by para from different things and then put together, and then say that you have finished your literature, review of literature. Try to understand what is published, try to put 4, 5, 6 things, 10 things together, collate the information, and have your own interpretation. That would be review of literature. And changing few sentences in between so that plagiarism software doesn't detect it will not really help you. If you want to build yourself, you must know how to really write. And literature collection is not outdated. Digital platforms, they provide opportunities to you, as I said, but that should not really be actually misused. So that's the kind of thing you have to do. And so without taking more time, there are teachers here, there are professors here, and guides here. So give enough independence to the students. That should be part of training actually. Our guides say, they will tell you something, but leave you, the whole world is open for you. You go with the idea, they will say, okay, go ahead, you do it. Not really fix in a frame, everything tight, and students can't move this way and that way and no ideas. And so, sometimes there will be not very important, brilliant ideas coming in, but it doesn't really matter. So, students should be given that kind of independence to explore their own ideas. And then only, their own potential which is there would be coming to the fore. Otherwise, it will remain hidden. So, how do we really do that? And that should be part of our teaching and training. How do we actually teach our own faculty to give independence to the students to explore the unexplored so that their full potential expresses? So frequent interaction is important to get out new things from the students, not to just give what you have and impose on him that he doesn't have any scope to explore his or her own ideas. So I think this is also an important component of our own faculties getting actually exposed to, and providing a catalytic environment for the best part of the students to be actually explored. I think this is what is very, very important. So I'm sure, and thanks to Dr. A.K. Singh, so we went around to the campus to see some of the new developments, and I'm sure we will be moving faster to develop the campus and to make a green campus. Dr. Agarwal keeps emphasizing, how do we make ICAR-IARI as a green campus? And if we make green campus, everybody has to, ride bicycles as we used to do, in 80s. And all the faculty and then students have to be ready for that. And not two-wheelers, And in the process, you will have a healthier life. And if you cycle, we used to cycle from ISRI to agronomy and engineering. And that, and there was no time, just, before classes, there is no time. Immediately after the previous class, you have to go to the other class. So, you can imagine, so, without proper breath and then breathlessly you have to ride cycles and reach the other division. So, that kind of situation should be enforced on you. Should be enforced on you. For your own health. For your own sake. That's very, very important. And today, in railway station, if you have to have a free pass, you have to go do some exercise. So that came in the newspaper. So, so if you have to get a free pass to enter the platform and in railway station, so you have to do some exercise, squatting exercise. So that kind of things have to be promoted in IRI campus. And those who don't come with their own vehicles and come with metro and then walk down and then walk here, they should be given some incentives. Okay. And in the campus also, those who don't walk, drive their cars and then bicycles. And how do we incentivize? Students can be given 2 marks extra for that. So some incentivization we have to do to start with. Not after 5 years, everybody will be falling in line. So anyway, so friends, all the best, and I have taken more time. And I'm sure Dr. Tushar Behra is now a proficient, molecular geneticist with his 1 year training in US, postdoctoral research, and all that. So that he would be actually leading, and Dr. Vishwanathan, I think we should be able to give them something beyond the course which is designed. So genomics, the translation part is fine, but maybe something more in theory if that can be given. So wish you all the very best. Have a wonderful stay, productive stay. I'm sure you will enjoy your learning experience here at IRI. IRI, a premier institution, we learn from here as students, and this particular program of National Higher Education project has given you opportunity. In all the training programs, we used to only call faculty members teachers. But Dr. Agarwal, thanks for providing this opportunity to the project, that now students are not only coming and taking training in the country, going abroad. Can you imagine 280 odd students going abroad, undergraduate students, going abroad to various universities and visiting and learning what is really happening there? So that is something which is remarkable in this particular program. Unparalleled. And I'm sure it will bring a sea change in our total thinking process and learning process. And I'm sure this program will also provide that kind of learning experience to all of you. Go more exalted, more excited, and I'm sure you will be learning much more than what you know today. And that will help you not only in your own thesis work, even beyond. I'm sure you will be able to get that advantage here because of the dedicated teachers, because of the well-designed course, and also, the new things that will be told to you. Wish you all the very best. Thank you, Dr. A.K. Singh, for inviting me.
Thank you, sir, for sharing with us your experience and for giving a message to our trainees that anyone can achieve success through hard work, determination, and courage. Everything that has beginning should end, and the end should be a happy one. And it is time to acknowledge those who have worked behind the curtains. So I request our Dean Ma'am, Dr. Rashmi Agarwal, to kindly give the formal vote of thanks.
Good morning, all of you. Our most respected and honorable Dr. Tirlochan Mahapatraji, DG ICR and Secretary here, who is the chief guest of today's function. Professor Chitranjan Kole, who is a Raja Raman Fellow and IPB and a very renowned researcher. Dr. E.K. Singh, our most respected director, and also Dr. R.C. Agarwal, a very dynamic leader, DDG Education at ICR headquarters. Dr. T.K. Behra, who is the course director of this particular training program, and also Professor Vegetable Science. I see the course coordinators, head of the division, Dr. Shaili Praveen and Dr. S.K. Singh and Dr. Vishwanathan. Other faculty members, students, ladies and gentlemen, and most important, the trainees. As very well said by our Honorable DG, that this is the opportunity, you have got it, and you will be learning few things in these few days, but it will open your mind. You can utilize the training given here for your not only the research program which we are doing, but also beyond. And sir, we are very, very thankful to you, sir, for accepting our invitation and giving us so much of ideas, not only for the students and the trainees and also for the faculty, that there should be a very, very nice relation between the student and the faculty, so that student is given free hand to do the research, give his inputs, and also give his thoughts to the research programs. So independence given to the student in his or her research work is a very important aspect. And the genomics area which you are taking up is a very complex area. And very rightly said by our Honorable DG that many challenges are there when you are working at genomics level, looking into the structure and functional genomics part of it. So all of our brains, so thank you again, sir, for coming here. I would like to thank our DDG Education, Dr. R.C. Agarwal, who is a leader and a very, very dynamic officer at ICR headquarters level. He is taking up all the higher education programs at national level, and you have heard that how he is bringing out the networking of all the universities and also encouraging the students at higher school level itself, so that they are encouraged to take up agriculture as their subject of choice, not as a subject when they are failing in other entrance examination and they are getting into agricultural field. So thank you, Dr. Agarwal, for your dynamic leadership and also providing all the financial and other support from ICR headquarters. Thank you once again. I would like to thank our Director IARI for all kinds of encouragement, support, guidance, and all inspirational words which he always keeps giving us, all the faculty and the students, sir. Thank you so much, sir, for giving all the support. Professor Chitranjan Kole, I said that he's a voracious reader, writer, editor, and a very eminent researcher, which is reflected by the publications which has brought out more than 100, roughly I think 130 publications on genomics itself he has brought out. So this is a very commendable job, and he has always been propagating the genomics research at the country level. So we are very thankful, sir. You came here and graced the occasion and said many words of wisdom to all of us. I would like to thank Dr. Behra. Very meticulously he has planned this training program, and as mentioned by many others, that horticultural crops were given a little back seat, but now many crops have been sequenced, and how these sequences can be utilized, how this can lead to translational research, and many other basics and the advanced practicals and the theory and lectures have been organized by Dr. T.K. Behra and his team, and dedicated faculty who will be taking lectures and practicals during the course program. So I am very thankful to all the dignitaries on the dais, and of the dais I am thankful to all the faculty, all the heads of the division who came here and graced the occasion, and most important, all the trainees. And once again, congratulations from my side that you have been selected for this very important training program, and this will definitely help you in your present research work and also in your future endeavors. With these words, I once again thank everyone and those who worked behind the curtain, stage managers and others who helped. So thank you once again, all of you. Thank you.
We have come to the end of the program. There will be a group photograph followed by a cup of tea.
Thank you, Anjal, for comparing so well. Anjal, you know, eh? Good.
also. Thank you, madam. Thank you, madam. Well, yeah.